It is estimated that more than 1 million people die every year due to drug-resistant bacteria. This happens when bacteria develops the ability to defeat the drugs designed to kill them and continue to grow until it kills the infected person. Resistant infections can be difficult and sometimes impossible to treat. It is considered by the World Health Organization as one of the biggest threats to global health, food security, and development today. This issue is increasing globally, and the death toll is just rising. It can affect anyone of any age in any country. We want to prevent this from being a reality for more people. But above all, we want to create a solution that goes with a vision towards the future challenges of healthcare. We wonder if there is something we can do a bit differently about antibiotics. Think about nature. We are not the best at designing things from scratch, but nature is. We don't want to reinvent the wheel, but to enhance it and direct it. Using bacteriophages, a biotechnology that has been silent for years, we can do this. There are viruses, just like viruses that infect humans, but these viruses infect bacteria and can cause their death. These viruses are very specialized structures and are delivered as targeted attacks to bacteria. To get deeper, we consulted experts like Marta Vives, a specialist in bacteriophages. With her, we understood that there are currently many restrictions on its use on humans, which causes a lack of research and hard regulations. Bacteriophages are so specialized that they connect with only specific receptors on the cellular walls. It is like receptors in the bacteria were padlocks, and the bacteriophages were the key to open that padlock. So they have to match. To address this, we want to leverage on technologies like AI to create such a special key. But the key must be somewhere in nature, and finding it naturally will take years. What if that key is within our reach of a few clicks? Having the ability to edit DNA in a specific way will allow us to have an infinite number of possible solutions for different infections caused by bacteria. With synthetic biology techniques, we can build treatments like Lego blocks, which can be mixed and matched between blocks of DNA from other organisms. We can cut out features from one organism and paste it into another. This technology is known as biobricks. With biobricks, we can lean on existing DNA in nature and reorganize it in other places. This tool is capable of making plants light up, creating perfume from bacteria, among many other things. All of this is accomplished from a protein code of adenine, cytosine, guanine, and thymine. Just as computer reads 1 and zeros to create images, DNA reads these codes to build everything we see in nature. By altering these codes, we can create infinite combinations within organisms. To better understand the subject, we investigated with Diego Hernandez, an expert in synthetic biology, with whom we carried out synthetic biology exercises using common bacteria to understand the procedures.
Hola, mucho gusto. Mi nombre es Diego. Actualmente soy técnico laboratorio en la Universidad de los Andes y me encargo de toda la parte de biología sintética dentro del programa de biodiseño de la universidad.
We wondered what will happen if we apply the same principle to viruses. Although viruses are not living organisms that made up of DNA, so like bacteria, they could be modified using the same principles. To address this, we are imagining the lab of the future, and we are calling it Synthetic Brown Lab, an all-in-one service that combines the capabilities of bacteriophages with synthetic biology and AI for the new antibiotic revolution. We are an integrated system that combines current technologies and powers them towards a future where creating an antibiotic from bacteriophages is possible with a custom-made solution in just a few minutes. With access to databanks, lab results, and the patient's clinical history, it is possible to decipher the perfect antibiotic match. To achieve this, Synthetic Round Lab relies on three main components. 1. A machine capable of reading AI predictions so it can edit the bacteriophage through a synthetic biology to create the personalized treatment. 2. An interconnected database with different hospital and research centers that contributes to the training and veracity to the AI to identify possible solutions quickly and effectively. 3. A personalized patient data interface that allows the doctor to make better decisions in a comprehensive way. Now, let's imagine a scenario of a synthetic RAM lab case. A patient is admitted to the hospital due to an apparently normal infection. The doctors evaluate him and decide to leave him in intensive care. The doctors decide to do multiple tests to find out what kind of infection it is. They take different samples, blood tests, and they suspect of a bacterial infection. A doctor requests additional FIN-Arrive tests, which is an automated pathogen prediction system, to start the protocol of synthetic RAM lab. After obtaining this result, it is processed by the AI using clinical information, symptoms, and hospital data to make a prediction. Based on this information, a better treatment, much more specific antibiotic is suggested. Later, we use a tool called the bacterial culture test to identify that the bacteria with the highest percentage of suspicion is the one that is causing the infection. When there is certainty about the bacteria causing the infection, the treatment to be followed is a multi-resistant one or is not. An alternative is provided to combat the outside antibiotics, such as bacteriophages. AI will show the studies about the combination that will work. Then, on the wing of the laboratory, the synthetic RAM machinery will perform in an automated way the bacteriophage and modify it until the desired one is obtained. When it's ready, it will be administered to the patient depending on the type of infection, whether oral, topical use, respiratory, among others. With this, we do not want to replace medical judgment, but to support the medical practice and decision making, but because we know in such a stressful environment that they operate with. This is why having the information at hand with synthetic RAM is able to provide the most relevant information to doctors, not only visually, but also through voice assistants, which allow them to hear the report with moving through the hospital. We know that there are many limitations for implementing this kind of technology today. It ranges from cost to legislation, but we propose a synthetic RAM lab as a possibility in the future where more studies with bacteriophages are really accessible technologies, and synthetic RAM lab can become a possibility in each wing of every hospital.